In case you don't know me, my name is Stephen Wingett. I'm a bioinformatician working in cell biology. I put together this talk because I think it's important for new students to know where to get started with processing and analyzing next generation sequencing experiments. The whole subject can seem quite impenetrable for the novice, but hopefully the next hour or so the next hour will go some way in dispelling some of the jargon associated with the field. So what do I mean by next generation sequencing? Well, you probably know, but I'll just sort of recap for those that don't. This is a newish technology that became widely adopted from around 2010 onwards. Next generation sequencing, or NGS, is a high throughput technology that allows for millions of different molecules to be sequenced simultaneously. It was a transformative technology, for its predecessors only allowed one sequence to be deduced at a time. This innovation, also known as massively parallel sequencing, enabled the development of numerous game-changing molecular biology techniques. So, for example, it made possible to deduce the transcriptome of a tissue, deduce protein DNA interactions, or indeed the methylation profile of a sample, among many, many other things. So that's all very well, but there's a bridge that needs to be crossed connecting the results that come off a sequencer to forming sound biological conclusions about the data. But fortunately, there's new methodologies and best practices that have been developed for the analysis of next generation sequencing experiments. So in this talk, we'll be looking at the initial processing steps, i.e. how to take files produced by a sequencer and convert these into data sets that can be easily interrogated to extract meaningful biological findings. So if you're planning to perform an NGS experiment yourself, I hope this talk is going to be useful. But even if you're not, you might find that during the course of your research, it sort of changes tack and you find that you actually do need to do some sort of NGS experiment. So hopefully it'll be useful for you as well. So my talk has three main parts. The first, I want to be talking about next generation sequencing. I just want to dig into a little bit about the technicalities of how it works. I think it's important not to just be doing an analysis with files and you just feel completely disconnected with the underlying technology. Then I'll be introducing the idea of bioinformatics pipelines, why they're useful, what they are, and essentially why you would use them. And then I'm going to get a little bit more specific about bioinformatics pipelines. And I'm going to be talking about the language Nextflow, which is used to write pipelines, and NF-Core, which is a suite of bioinformatics pipelines that are publicly available. Okay, so next generation sequencing, what is it, or its applications and data types associated? So as I alluded to a little bit before, next generation sequencing is a massively parallel sequencing technology. I guess the best known is the technology produced by Illumina. That's a company, well, it's a company. The actual technology was developed here in Cambridge, but then it was bought by Illumina, an American-based company. And this technology distinguishes itself by being very high throughput. Okay, you can literally sequence millions or billions of reads simultaneously. I guess the two main competing technologies are those produced by Oxford Nanobore and PacBio. And they're lower throughput than the Illumina technology, but they distinguish themselves by producing longer reads. So Illumina would typically produce reads of 100, 200 base pairs in length, whereas these other technologies, we can be talking kilobases or indeed even megabases in terms of length. So that's really the sort of landscape of next generation sequencing. As I said before, it's been a transformative technology. It's either completely sort of revolutionized existing fields or it's developed new fields. So for example, whole genome sequencing, you can now sequence in an organism in a day or two, whereas it would take months or years. You can identify the transcriptome of an individual tissue by RNA-seq, or you can identify, for example, protein DNA interactions using ChIP-seq. You can look at within chromatin regions of accessibility or regions which are more closed using ATAC-seq. And then you can perform targeted sequencing technologies using things like exome sequencing or targeted gene sequencing. And even in the field of epigenetics, it's been useful where you can, for example, use techniques like bisulfite-seq, where you can identify the methylation profiles of CPG islands in the genome. Metagenomics as well, so the idea you could go out on a boat, fill up your collection vessel with seawater, extract the DNA and then sequence it and work out all the organisms in there next generation sequencing's made that possible as well. And also, maybe more surprisingly, at its inception, it's helped elucidate the three-dimensional structure of a genome. So with chromosome capture technologies like Hi-C, you can sort of build a sort of three-dimensional structure of a genome, and that sort of helps elucidate the function of a genome. So you may have, say, for example, have an enhancer and a promoter, which seem quite separated when you just look at the raw sequence data. When you look at this in 3 dimensions, you realize they're actually quite close, and that sort of hints at their functional significance. So NGS has been transformative. I probably should just distinguish at this point the difference between single cell and bulk sequencing experiments. As the name suggests, single cell experiments allow you to perform the sequencing, but then at the end you can work out from which individual cell a read was derived. Bulk is the same, it's sequencing, but you don't have this single cell resolution. So bulk is easier to perform. It was the pre-existing technologies you'd probably expect, and it's cheaper as well. So that's why for a lot of applications it's fine. It allows you to identify, for example, for RNA-seq, bulk RNA-seq, you could look at a tissue and you can see one gene is highly expressed and another's are lowly expressed. Or you could draw comparisons between different samples. So you might identify genes which are differentially expressed. So it is useful, but it does give you global averages. So if you imagine you've got a tissue where you see a gene that has some sort of mid-level expression, that might not give you the complete picture. When you actually look at the single cell population, you may see for that gene, half the cells in the population express the genes at high level and the rest expressed it at low level. And this sort of mid-level expression was not the true picture of any of the individual cells. So single cell sort of sequencing helps reveal this cell heterogeneity within a population, which is useful. It's also good for identifying sort of rare cell types that you wouldn't always notice in a sample or tissue, or indeed even transitions from one cell type to another. So it's an important technology. So it's quite well established now, NGS, in terms of actually performing it. I mean, I'm not a wet lab person myself, but I get the impression you create your library and you can pretty much put it on a sequencer and you'll get the results out the other end. But there are different types of sequencer, and that is something to consider. So these are just examples of Illumina sequencers, and on the left-hand side you have the more low throughput machines, and on the right-hand side you have the high throughput machines. For example, the iSeq produces 4 million reads, whereas the NovaSeq produces 20 billion reads. So there's a huge range, and you need to decide before you're sequencing what exactly do you need to get out of the experiment. Obviously, you don't want to spend a lot of money on the very expensive high throughput machines if you only need a low sequencing depth. But conversely, you don't want to be performing sequencing, at a very shallow level, and then the results are actually meaningless. So you do need to think about sort of bioinformatically what you're going to be doing with it later on. Maybe the best way to deal with this is look in papers to see what people have done before to try and work out the sequencing depth you need, and talk to the sequencing facilities and bioinformaticians. But that's essentially how the sequencing works. I think we've got a Nextseq here at the LMB. I've lost touch a bit with the sequencing guy who's stuck to his left, but I think we had a MySeq and a Nextseq, so it's sort of in the middle there. But yeah, sometimes if people outsource the sequencing, they sometimes use NovaSeqs and outsource it to a company or to Cancer Research UK as well. So yeah, that's sort of how it works. So these sequencing machines, they're impressive, but it's sometimes just easy to think about them as this sort of magical black box. You put your sample in and you get sequences out the end. I think it's good to just try and understand the molecular biology that's going on and how the sequencing works to sort of bridge this gap. So the sequencing is made possible by these, essentially these Illumina adapters. So you have your sample, so that's the DNA insert you can see here that you're interested in sequencing. And what you would do is you would ligate onto either end these Illumina adapters. And that essentially is what would constitute your Illumina sequencing library. I don't really want to talk about this slide in any more depth than that, except it's these adapters that make the sequencing possible. So what would happen, when the sequencing takes place, there's 2 main steps. And the first step is something called cluster generation. And this takes place in your sequencer. So you sort of open it up and there's a cartridge, and in there will be a flow cell. And then you would load your sample into one of the lanes in the flow cell. Now, the secret of this technology is this oligo lawn. So the flow cell is coated with oligonucleotides, which are represented here in these, I think they're green little circles here. So these are individual nucleotides, the dots, and you can see the oligo lawn. And what will happen is your DNA strands that from your library will come in here and they will hybridize to the oligo lawn. So there is Watson-Crick base pairing between this strand here and the oligo lawn. So that's the Illumina adapter. It's complementary to the oligo lawn. So that's how it attaches itself. And then what you do is you perform an extension reaction. So you'd add a polymerase and you would prime off the oligo, and this single-stranded DNA molecule would become a double-stranded DNA molecule like so. Then what you do is you denature it. That original strand would float away after the denaturing. And what would happen is you've just got now a single strand of DNA, which is covalently linked to your flow cell. So it's tethered there now. Then what would happen is you've just got to think, sometimes you can think of these molecules as quite static, but they're not. They're sort of moving around at high speed. And invariably the other end, which has another Illumina adapter, will then hybridize to another oligonucleotide like so. So you get this structure here, which is tethered at one end, and then there's this Watson-Crick base pairing here. And then what you do is you perform another polymerase reaction, and you now have double-stranded DNA. And then you denature it once more, so you end up with two strands which are tethered to the flow cell. You perform another replication reaction, and then you double it up. These will then form these bridges again, and essentially the whole thing will just expand outwards. So essentially what we're doing, it's called bridge amplification, but what we're doing is essentially a PCR reaction. But unlike a typical PCR reaction where the molecules and products are free in the solution, they end up being tethered to the flow cell. So you end up with this cluster of identical DNA molecules on the flow cell surface. Okay, so that's how it works. And you've got to imagine on the flow cell there'll be other molecules which will seed the flow cell and form their own individual clusters. So I guess it's a bit akin to in bacteriology, where you do serial dilutions of bacteria, put them on an agar plate them out, and then the next day you've got a colony of bacteria. Here you've got clusters which were created by individual DNA molecules. Okay, so that's the first part. The second part of the Illumina sequencing is very much like the conventional Sanger sequencing. So the idea is you'd have your single-stranded molecule here, which is tethered to the flow cell. And what you would do is you would add nucleotides, which are labeled with different colored dyes, and they have a reversible terminator on them. And what would happen is you add your polymerase, and it will start to extend the molecule, but the reversible terminator will mean only one base can be added at a time. So this base will be there, and it will have a dye attached to it. At that point, you photograph the flow cell using a microscope, and you shine a laser at it, and the dye will emit a color, and the color will be dependent on the base which is there. Okay. You then remove the reversible terminator and add the second base, shine a laser at it, photograph the flow cell, and then you've got a second image. So essentially what you end up with is like 100 images of the flow cell. And what you can see at this point are the individual clusters in each image. And each of the different images will represent the first base, the second base, the third base, the fourth base, and so on. So what you can do is you can look at the cluster, see how it changes through each of the individual images, and that tells you the actual sequence of the DNA molecule. Okay, so that's the sort of sequencing technology. I just wanted to introduce a couple of terms as well, which crop up sort of endlessly whenever you talk about sequencing. There's single end and there are paired end sequencing reactions that you can perform, or sequencing runs. Single end, what happens, you've got your DNA insert of interest, but the read that you get back will just come from one end. So it'll just be reading in this 5 to 3 direction. The paired end reads, it's a little bit different. You have two reactions which are taking place. One reaction will read the insert from one end, and then the other action will read the DNA from the other end, like so. And these two reads may or may not overlap. It just depends on the length of the DNA insert and the length of the read. So that's the idea. Just a final little bit of terminology I wanted to talk about. So in sequencing experiments, sometimes you can add barcodes. So these will be an additional read, which is typically about eight base pairs in length. And what they do is they allow for multiplexing. So the idea is you would load multiple samples into the same lane onto your flow cell. You'd perform the sequencing reaction, but associated with each read will be a barcode read. And then what you can do is when you've got your data file at the end, you can allocate each of the individual reads to an individual sample. So this helps you save money, essentially. If you've got a NovaSeq waiting to go and you've got your sample, you don't have to have one sample per lane. You can put all your samples or 96 samples in your Illumina NovaSeq, and then at the end you can demultiplex them. So that's useful. Just another terminology that crops up quite a bit are unique molecular identifiers. And these allow you to distinguish between genuine biological duplicates in your sample. So these are DNA molecules which have come from independent biological sources, or PCR duplicates. So PCR is this method, as I'm sure you all know, of amplifying DNA. Unfortunately, it tends to bias some sequences more than others. And what you can end up with is when you look at the results, you could see maybe a few sequences predominating, and that's actually an artifact of the PCR reaction rather than being present in the initial sample. So by looking at the unique molecular identifiers, these are barcode sequences. They're about, I don't know, sort of 26. One example is a 26 base pair barcode, and they're random. They're random sequences. So what you wouldn't expect is the same molecule by chance to have the same UMI. If a molecule has the same UMI, you can be sure it's a PCR duplicate and remove it. Okay, so that's the idea. I think I wanted to talk a little bit about the sequencing technology, because before you, whenever you perform some sort of sequencing run and you then got to analyze the data, it's important you understand exactly what it is you're getting back. So you need to know sort of where, for example, maybe the results you get back won't begin with the sequence of your interest from the genome. You don't know you need to know how many reads you're getting back. You need to know how the reads relate to one another. So it's really important you understand the constructs before you sequence them, so you know sort of the correct analysis that you need to perform. So yeah, don't just think of it as two disconnected universes. So that's really what this next part of the talk is about. I've talked a little about the in vitro situation. You've prepared your sample, you've put it on the sequencer. The sequencer's run. What do you actually do? What do you get back? What do you do with the output? Okay. So what you actually get back for almost all sequencing runs are FASTQ files. So they sound like quite fancy, but essentially all they are sequence text files. So you could open them with Notepad if you needed to. Typically, actually, they are a bit too large to open with Notepad. But the idea is, it was just like 1 million reads, you could open them with Notepad. Having said that, they're normally compressed. So I guess most people here are familiar with the idea of zipping large files. These are very large files which can be gigabytes in size. So what we use is a program called, well, a zip program. It's actually called gzip. And you'll get these gzip text files coming out the other end. And what you'll see when you look inside these files, these FASTQ files, is text that will look like this. So this is one read. And it's just one read. And what you can probably expect is that's the nucleotide sequence of the read. So it's literally just the AGCT sequence. If there is any ambiguity regarding a base call, you'll get an N in there as well. So that will be the 5th letter. So what are these extra lines? You've got a row of C's here. Well, this is actually a quality assessment of the base calling. So we're saying that this T was called with a quality score of C, this G with a quality score of C, and so on. In fact, everything was called with a quality score of C, apart from the penultimate thymine here, which has a quality score of B. And I'll talk a little bit more about how they're derived in the next slide. Also, by convention in a FASTQ file, you'll see this plus. It is just by convention, so it's just there, so you can ignore it. But the header line is sometimes of interest. Often you don't need it, but if you need to dig into your data more, maybe for troubleshooting, understanding what's going on, it does contain valuable information. It always begins with an @ Yeah, it will then have the sequencer ID listed, or if it's from an external repository, it will have the name of the sequence accession, which is what you can see here. And then it will have other information such as the flow cell ID that was used, the lane ID on the flow cell that was actually run, and then it has a grid reference for where the cluster for this read was detected. And then after that, it might give you other information. So here it tells you the length of the read was 40 base pairs. So it's important to really try and demystify what these are. They really are just files with nucleotide sequences in that you get off the sequencer. When I mentioned the quality scores earlier, and we had these row of C's and then a D, all they really are numerical values. And we have a code beginning at exclamation mark and ending at I. So that is a quality score of 0 if you have an exclamation mark. And if you have a score of I, it's 40, and then in between it's 1, 2, 3, 4, 5, and so on. I don't know if there's any sort of computery people in here. That's actually underpinning this. It's like the ASCII code, numerical representation of characters. But really, you don't need to know that. You just need to know that it's just a numerical value. And technically, that's defined as the probability of a base call being incorrect is the p-value, and then we do the -10 times log 10 value of that. So if we said that the probability of this base call was incorrect, we would have a p-value of 1, log 10 of 1 is 0, and then we'll end up with a quality score of 0, which is exclamation mark. So this information, the sequence and the quality scores, are used by downstream tools for the analysis of the data. So this FASTQ format is basically your sequence with quality scores. Okay, so I mentioned FASTQ files. So I just want to take a little bit of an aside here to bring in a note that's a bit of my bugbear. But I think if there's anything that, I mean, I'm going to be droning on for an hour, but if there's one thing in this talk I would like you to take away and remember, it's just about data storage. I know this is a bit prosaic, but whenever you do a sequencing run, download the data promptly from the sequencing facility or the in-house sequencer. It can be deleted after a month. So if you forget about it and then do it last minute, and then you think, oh, hang on, it's not quite worked, you can lose your data that way. Also, what people don't realize is these files are very big. So they can be tens of gigabytes in size. And when you download them, they can get corrupted. So they might just get truncated, which you might spot if you notice the file size is a lot smaller, but you might not. Or sometimes the data internally can get corrupted. So you end up with the right file size, but it's actually garbled in the middle. And you've got meaningless data back. So this is a bit of a disaster. But there is a way around it. The sequencing facility will provide you with a text file with something called MD5 sums. So it's just a fancy term, but an MD5 sum is just a little calculation that's performed on your FASTQ file, and it will summarize your whole file as like a 20-character string, okay? And what's important is if a FASTQ file just changes by 1 bit, that MD5 sum will change, right? So, and just by chance, by the length of it, no, you wouldn't expect any FASTQ file to share an MD5 sum with another FASTQ file. So what happens is they will run their MD5 sum algorithm on your FASTQ files, and they'll send that to you separately, or you can get hold of the MD5 sum files. Then you run the MD5 sum program on the FASTQ files on your server, and if they match, you know the download's worked. Always do this, because it does happen quite a lot that people download their data and it's actually garbled and they don't realize. It's important because FASTQ files can't be replaced. So if you've gone to the effort of doing the sequencing and they've gone missing and you don't realize afterwards, all of that work, all of the sequencing, maybe all the lab work has gone to waste and you've got nothing to show for it. So make sure you copy it to a secure backed up location. Typically, that would be on iStore or iSalon, but do check with your group leader where you store them in your group. And if in doubt, speak to IT or scientific computing about that. So do get that right. Is anyone in cell biology here? Yeah. Okay, so we've just started our own dedicated storage space in cell biology. Tim and I managing that. So if you provide us with your FASTQ files, it will be stored in-house. It's also backed up off-site at the Crook, and every 6 months we back it up to tape. So if you give it to us, it should be, you know, absolutely secure, barring some, I don't know, something terrible. I mean, have your own copy as well, but we are sort of rolling this out now. It's got its own GUI and front end, so you just give us a metadata. There's like a form that takes 15 minutes to fill in, and then you and your PI will have access to the files that you deposit. Okay, right. So you've got your FASTQ file. So I guess the next thing you need to think about is what is it that you actually need to do next. And this can be a little bit daunting. You've got these files of letters. What do you actually do with them? Well, first you need to think about the type of experiment that you've performed. Is it a standard typical sort of next generation sequencing experiment like RNA-seq or ChIP-seq or something? Or is it some sort of different bespoke experiment that might need some sort of customized solution? If it's sort of more standardized, there are ways to deal with it. If it is something off-piece, you might actually be having to get into the world of having to do your own code and try to analyze the data that way. But most people who do sort of sequencing experiments at the LMB can be accommodated by the pipelines that I'm going to talk about next. So whenever you do an experiment, you need to think about what is it I'm trying to do, and then how do I do it? So what software do I need? And there's probably going to be multiple steps in your analysis. So there might typically be some sort of mapping step. And then when you've done that, you'll pass these files to some other tool and they'll have to process them. And you'll have a whole plethora of tools that need to be used. So just to maybe give you an example to flesh this idea out, it's normally, for most next generation sequencing experiments, one of the key steps is to map the data. So the idea is that you've got your sequence reads and you want to understand to where those reads were derived in your organism, okay? So what you would do typically is you would get some sort of mapping tool. So Bowtie 2 is one of these. There are other tools called BWA, STAR, and so on. And what you do is you provide it with your FASTQ files, and you would also download some sort of reference genome in FASTA format. And you would pass this to your mapping tool. And then what this tool will do is it will work through your FASTQ file, sequence by sequence, and it will work out best fit to where that read came from. So if it doesn't work, it'll throw it away, but it should be able to work out from your sequence the location in the genome where that read came from, okay? And then what that will do is it will collate the data and it will output it for you as something called a BAM file. So we've started with a FASTQ file, we've ended up with a BAM file. So this is how these software will create data dependencies for you and additional data. So let's have a look. So what's a BAM file? Well, essentially it's just a binary version of something called a SAM file, which is a sequence alignment map file, okay? To process BAM files, you need an additional tool called SAM tools. So we're starting to build up a network of tools now. We need to actually do anything with our data. But if you look inside a SAM file, it sounds a bit daunting again, but it's actually quite easy to understand once you know how. At the start of the file are these header lines, and they just have @ always at the start. And then what you have is tells you a little bit of the version of the software, and then it tells you about the length of the chromosomes or scaffolds. So it says we map to chromosome 1, chromosome 10, chromosome 11, and so on. And chromosome 2 will be included in there as well. It's just the way it's sorted. And then when you get past the headers, you'll get through the information that tells you all about the read alignments. So some of it will look familiar. You'll have in the red, you can see your sequence, and that's ported over from the FASTQ file. And then you can see the quality score. And then here you can see the header from the FASTQ file. And there's other sort of metadata here that's interesting. But key is the thing I shaded in purple, which tells you the chromosome to which this read mapped and the position, okay? And then for every read that you map, in your BAM file will be all this information, and it will contain the location to where the read mapped. So that's the BAM file. That's what you've made by the mapping process. Okay, so that's fine. But what you can start to think of is here, okay, I'm starting to generate more files, and you'll see on the next slide, you're going to generate even more files in a typical workflow, and you need to think about how best to manage that. So if we go to the next slide, I give you a simple example of we've got a FASTQ file and we want to map it. But in reality, that's not all you want to do with the FASTQ file. What you'll need to do is do some sort of quality control on your file. So that's just checking to make sure the sequencing worked, the data looks as it should, there are no sort of files which have been duplicated or anything like that. You'll need to check that. You should always check this. And you'll need to do the mapping, of course. But actually, for good sort of practice, before you do the mapping, you want to remove reads which are low quality. You want to get rid of those from your data set. And indeed, there might be reads which start off towards the start of good quality, but end with very poor quality. So before they're presented to a mapper, you want to trim them. So there's a trimming step. And then normally when you've got the BAM files out of the mapper, you want to do something with it. So for example, if it's RNA-seq, you might want to align the reads that you've got out to a reference genome so you can quantitate the expression levels of individual genes. So you'll need a separate tool that performs the quantitation for you. So you'll need to handle this process. And then at the end, you'll probably want some process where you can take all the summary reports from all these files and collate it so you can have a global picture of whether everything has worked. So this is quite a time-consuming task. And there are a lot of steps to doing this. And sure, you can just do it step by step yourself, but that's not how people tend to do this anymore, or bioinformaticians tend to do it. So what would happen in the past is, as a workflow becomes a little bit more standardized within a lab, a bioinformatician would join the software together with custom scripts. So this would take the output from one bit of software and pass it to the next bit of software. And that sort of worked fine, sort of for a while. These scripts were typically written in Python or Perl. But the problem is that with these languages, they weren't geared specifically for handling data flow through pipelines. So the sort of computer programmers, what they did is they developed a new bit of software, well, a new coding language, in fact, that can handle running the execution of software and the execution of software on different types of computing platforms. And it also manages for you as an inbuilt property the data flow through the pipeline. And the one I use and I've set up at the LMB is Nextflow. There are other rival pipelines for bioinformatics. I guess SnakeMake is the leading contender among these. But that's essentially where we are. We have a new programming languages for managing data flow through pipelines. So that can all sound a bit daunting and a bit computery. While you do need to program to be able to write your own pipelines, you don't need to be able to program to actually run existing pipelines. So if you've got a pipeline that's ready to go, you just need to learn a few commands to be able to run them. And once you do run them, all the sort of the steps that I showed on the previous slide of the QCing, the mapping, that's all managed for you as part of the pipeline. And you can perform sort of multiple or hundreds of tasks simultaneously. So people ask, once this is set up, where is the best place to run a pipeline? At the LMB, I noticed quite a few people do try and do some analysis themselves on their Macs or on their PCs. I know people have got sort of nice, sort of expensive Macs and they're quite powerful. Really, the best place to do it, however, is on the computer cluster. So we've got a high performance machine at the LMB that's managed by Scientific Computing. It's got an enormous amount of memory in terms of storage. It's also got enormous RAM, and it's got huge throughput in terms of these individual nodes that can process jobs simultaneously. So if you've got to handle these large data sets and process them in a manageable time frame, I really would recommend getting to grips with a computer cluster. Just a bit, does anybody actually, just what I mentioned, does anybody know how to use Linux at all or use a cluster? We've got one person. Okay, there's some wavy hands, so I'll mention this. If you are familiar, if you use Nextflow on a cluster environment, it manages the job submission for you. So what it does is it runs as a tiny little daemon process on the head node, and then it creates SBatch scripts for you. and then it executes them, and those are what are used to submit jobs to the compute nodes. Okay, and then when one job is finished, the new SBatch script will be generated, and then that will be sent out, and so on. So it's it's good to know Slurm, but you don't actually have to know it, in any great detail to be able to run a Nextflow pipeline. So that's fine. Bioinformaticians and computer people have developed their own programming language to handle pipelines, but we still had a situation where bioinformaticians were turning up in new labs. They wanted to do a routine bit of work, and what they ended up doing was writing their own script again, and then running it. And then what would happen is that bioinformatician would leave, and then another one would come in, and someone would write a new script, which is, it's great for keeping you in a job, but it's not ideal. So what this sort of reinventing the wheel that was going on, people had recognized this, and they realized it wasn't efficient, and it needed to come to an end. So a community of developers got together, sort of experts in their field, and they developed a range of pipelines which are freely available. They're open source, and they're available, and they constitute what is known as NF Core. So there's about 80 pipelines or more available now, and they accommodate most sort of typical bioinformatics sort of analysis. So RNA-seq, ChIP-seq, all that sort of stuff, and so on. They're a good community. They're pretty well documented as well. So if you need to understand how they work, you can go in there and read about what's going on. There's also the community is available on Slack. So if you've got questions, you can ask people who develop the pipelines how it works, or help you troubleshoot the situation. They also, if you want to get into using NF Core, they do tutorials in town and at the Sanger as well. So that's a good place to go. Also, what's very important is, and useful, when it comes to writing up a paper, these pipelines are highly reproducible. So if you run your data on a pipeline at the LMB, you should get the same results if you run that same version of that pipeline somewhere else. And these pipelines are all under version control. When you come to write up your paper, you said, I did, the RNA-seq mapping. You just said, I use Nextflow RNA-seq pipeline version 3.6. People will know how to do that at the other end. So it's very useful for materials and methods. They used at other institutions as well. So they used at the Crook, the Sanger, the Crick as well, CRG in Barcelona. So they're used at the sort of noteworthy institutes across the world, and many others as well. And that's the web address there. Okay, so I just wanted to just briefly summarize, just give an overview of a pipeline. This is produced by NF Core. This is the consortium that produces these pipelines, and this is for ChIP-seq. And what they do is they produce these little tube maps to try and describe the data flow through the pipelines. So you can see you have your FASTQ file that comes in, then there's a bit of quality control mapping. There's a few other steps around here. And then based on the map data, you do peak calling with max3. Then you sort of, generate consensus peaks here with Homer and bed tours. You can then look for differential peaks with R and DEC2. And then you can summarize all the reports with multi-QC. So that's the sort of thing that they present. And then each of these different steps, there will be little parameters typically to alter how they work, but normally the defaults tend to work fine. So that's a pipeline. I have set up a number of pipelines already at the LMB on the computer cluster. I've installed one for downloading FASTQ files. So if you need to go to a public repository and download the FASTQ files, you can just put in an accession number and it will get hold of it for you and download it, and all the metadata. That sounds trivial. Actually, sometimes these external repositories have these sort of labyrinthine menus that it's actually quite hard to get hold of your data, but this helps. There's a pipeline for QCing your data. There's also an RNA-seq, ChIP-seq, and a TAC-seq pipeline on there. So if you do those experiments, you should be able to get going quite quickly to get results. There's also cut and tag, single cell 10x pipeline setup. I've also written a pipeline for the parse Evercode single cell technology. So if anybody's doing this new sort of split-seq technology, I have put together a pipeline there. And there's also a pipeline for taxonomy profiling. if anybody's got any experiments that they're going to do that aren't covered by this, and we can see them on our NF core, I can try and set them up for you. Sometimes it does take a little bit of sort of, you have to sort of tempt the cluster to actually get it working. But after a bit of troubleshooting, they do tend to get set up. So just come to me if you need any sort of pipeline set up on the cluster. To actually be able to run these pipelines yourself, you'll need to be registered on the cluster. So I think there's an online form by Scientific Computing. If that doesn't work, go to Scientific Computing. But just as a bit of upfront knowledge, you do need to know how to use Linux and the command line. And it is sort of useful to have to know how to use Slurm, or at least what it is, this idea of scheduling jobs to a cluster. And you also need to install Nextflow as well. So I know that can sound a little bit overwhelming. As a QR code there, I've made resources online available for how to use the cluster at the LMB. I keep that up to date. It's part of a GitHub repo, and if there are changes, I will change that. I also teach this course twice a year, so there's like questions and exercises, and it helps people get started. So yeah, if you need to know how to get set up on the cluster, that should help. And talk to Scientific Computing or me as well if you get stuck. There we go. Okay, so the commands that you send to the cluster can seem a little bit daunting. This is a typical Nextflow command, and this will execute this NF core job here, and it's an RNA-seq pipeline, version 3.6, and you have to specify a design file, you have to specify the genome you want to map to, and then there's this config thing that you always have to put in, specify your results file, and so that can look a little bit overwhelming. It's like, I'm not really that computer familiar with the pipeline, and what defaults should I use, and so on. I have tried to make it easier for people at the LMB. So I put together a little, so this is actually running off a local host, but the internal web link is GuidePiper. And what you have is a list of all the pipelines I put together. And you just pick the one that you want to choose. So I don't know, if you had RNA-seq is probably the most straightforward. And there are just some drop downs that you need to do to get the command to run your pipeline. So, for example, you're going to map to human. You just set the human genome, wherever that is. If it's there, yeah, there it is. Then there's a little design file that you need to put together. So this just tells you how the experimental FASTQ files relate to one another. So if you do test.csv, for example, and then it just asks you whether or not you use a NovaSeq. So let's say we did. And then we generate a cluster command, and we generate the command that we need there. So you just do some drop downs, you'll get a command, and then you just copy and paste this into the cluster, and it should kick off. It should perform all the jobs that you're doing, all the mapping and everything, and then it'll send you an email when it's done. So that's how I've set it up. Okay. Yeah, that's the link, GuidePiper. Okay. So just a few notes on running the pipelines. One of the things, the sort of gotchas that catch people out, and you really want to avoid, is not checking the quality control reports when they come out. I know you've done a fancy experiment and you've got data that you actually want to make biological sense out of, but it's what we were saying before. If you've got garbage in, you're going to get garbage out. So so with the pipelines we've put together, there is extensive quality control produced. So this is produced by a well-known tool called FastQC, and it gives you an overview of the quality scores of your data set. So what we have are individual box plots, and each of these boxes, well, it's a box plot, and each of these boxes represents the quality scores at each position within a read. So the first nucleotide has a good quality, and then you can see the quality score starts to disintegrate a little bit as you go along the read. Actually, this is a good quality run. But imagine if all your sequencing data were in the red here with very poor quality. I'd really think very carefully before you actually go ahead and analyze this data, because it's probably going to be rubbish. So that's the sort of thing you need to check. Likewise, another key quality metric you'll get off a pipeline is the mapping efficiency. So STAR is a commonly used aligner. It's a splice-aware aligner for RNA-seq. And what you'll get from the pipeline is a little diagram like this. And you can see in blue the number of reads that mapped and in red, the number of reads that didn't map. And this is quite good because most of the reads do actually align to your reference genome. If they were all red, don't go ahead and start analyzing the expression matrix, because it's probably just artifacts and rubbish. So definitely do check this whenever you're doing anything. So yeah, from the pipelines, typically what you will get out the end are copious QC reports, which you should check. Often there'll be some sort of quantitative data, and that'll be the raw data. It'll be normalized in some way. So for RNA-seq, you would have a gene expression matrix, you'd have the raw count, maybe the TPM values, and so on. Well, you will get the TPM values and other normalized of counts as well. And you might, for other pipelines, you get other sort of metrics, like you may get peak positions for a tag-seq or chip-seq, cut and tag, and so on. And you get nice little features as well. Like, for example, if you want to view your data in the IGV browser, you get IGV sessions all set up for you, so you can go ahead and have a look at your data as soon as it comes off the pipeline. So that's the sort of things that pipelines can produce for you. Just at the end, so you've done all your work, you've done all your analysis. I just wanted to mention again, when it comes to submission, you do need to provide FASTQ files. This is probably my second slide that I want you to take away. You will need those original FASTQ files. You don't want to lose them. You don't want to be in a position when after 4 years, you've done your sequencing, you've done your experiments, you've done all your analysis, you come to write your paper, and then Cell or whatever will say, deposit your FASTQ files on an external repository. You've lost them, and then that's well, we're not going to publish any of your work. That is what will happen. So look after sequencing data like gold dust. Okay. So yeah. Okay. So that was a bit of a whistle-stop tour through pipelines and NGS sequencing, but we've covered quite a bit of ground. We've looked at how Illumina sequencing works. I just wanted to make sure people sort of have an idea of the sequencing methods. And we've looked at how the sequencing that's performed off on a sequencer gets translated into actual data files. So these are the FASTQ formats. And then we introduced the idea of software that's going to be used, such as aligners, to convert FASTQ, raw FASTQ reads into some other data formats, such as BAM files or expression matrices, and so on. And then I introduced the idea of joining these different software tools together using pipelining language, such as Nextflow. And not only that, not only can you write pipelines using Nextflow, there's already a whole range of pre-compiled, pre-produced pipelines produced by a consortium of experts at NFCore, which are available for you to download. Well, they are available to download, but you can have a go at using some of these yourselves on the cluster. And I've given you a little bit of introduction on how to run the pipelines as well. So there's just one final slide. It's a bit of a plug again. I mean, I've been speaking for an hour, and I've tried to give you a flavor of pipelines and how you use them. I'm not expecting you to be able to go away and actually run these pipelines yourself. I mean, great if you can, but again, there is a second course I put together, which is especially, so it assumes that, so if you've done the cluster course, you can go to here. It's updated like before, it's on my Git repo, and it talks you through the process of getting set up on the cluster, installing Nextflow, how to run an NFCore pipeline, and then it talks you through the output and so on. So that's the second course, and hopefully between this talk and the 2 courses I've made available, you guys can have a start at doing any analysis yourself. So that really is it. So thanks for listening.